And we are live again. So, back to our big triangle pyramidal cell and our main player over here. So I kind of switched the color of the mitochondria, but you still get the impression this is navy, not black. But that's our mitochondria and our endoplasmic reticulum. Oh, that changed color too, but whatever. Endoplasmic reticulum buffering calcium. So high levels of intracellular calcium, we're still using that as our starting point in this whole process. The excitotoxicity that's produced by activation of those NMDA receptors. Mitochondria now helping to generate ATP to keep that endoplasmic reticulum membrane intact. So I remember I talked a little bit about the membrane. There's channels in here that are meant to keep that calcium buffered within the endoplasmic reticulum, and those channels need to be held closed through the generation of ATP, the energy. It's an active process. So the more calcium you have in, the higher levels of ATP that are going to be generated to help buffer that calcium. So what does that do? So what we talked about in the last little video segment is that those mitochondria, that ATP, or one result of that oxidative phosphorylation reaction is to generate oxygen or reactive oxygen species. Okay, that's not exactly it. But the point is you're getting these reactive oxygen species to start kicking out, and they are going to break down the cell membrane. So let me just review those quick. Superoxide ions, O2, hydrogen peroxide, and hydroxyl radicals. And they break down, as they break down the membrane, that sets off this process of lysis. So the cell membrane breaks down, and that can lead to another process, which we will get to. No, that's not going to show up well. Let's just make it in red. Neuroinflammation, which is a really common feature in a lot of neurodegenerative diseases. But another common feature is apoptosis. So that's what we're going to talk about here. So I'm going to put it outside, even though it's happening within the cell. So again, the mitochondria, as they're working, building up, producing this ATP through the oxidative phosphorylation process, what happens is they start to kick off these signals. So this is all, again, inside the cell, talking about apoptosis. They kick off what are called Cytochrome C Enzymes. So those enzymes are normally useful to keep these reaction processes going. So Cytochrome C. Okay, that's a signal that essentially, or a post-translational modification or an enzyme or reactive species that turns on or activates another protein called caspase. So as cytochrome C cytochrome C is coming out of the mitochondria, still within the cell, so just make sure you get that it's within the cell boundaries, within those cell limits. So all happening inside the cell. Cytochrome C is coming off, activating caspase. So there's lots of different species, caspase 3, caspase 9, it doesn't really matter at this point, but caspase is a signaling molecule that can now feed back to, so again, remember we're inside the neuron. This will be our nucleus with our double-stranded DNA. So that's what's being drawn here, double-stranded DNA. Let's make a nice distinct color. I don't have much. Here we go. It's a double-stranded DNA. That didn't work out well. So caspase will actually signal now into the nucleus and set off apoptotic signals. Apoptosis. So it's programmed. This happens during development. You're going to be able to see that. I hope so. Programmed cell death. This is a normal process within the cell. During development, it happens all the time, right? Because you want to lose some of your cells. You're born with an overabundance of neurons. So there are signals within your DNA that can turn on apoptosis and lead to programmed cell death. So what that means is that so let's go back to our on the aging side now. So you have these neurons that are vulnerable because there's less ability to buffer calcium. So there's more calcium coming in for whatever reason. Maybe there's extrasynaptic calcium, or extrasynaptic, actually, glutamate, meaning not synaptically related to the release of glutamate that often helps you learn and remember. So extracellular, extrasynaptic glutamate caused by a stroke, caused by a brain injury, caused by whatever, too much alcohol, can lead to high levels of calcium within the neuron. And the endoplasmic reticulum can buffer that up to a certain point. But slowly, it needs more energy. So the mitochondria start generating that energy through ATP, through those oxidative phosphorylation reactions. As they generate ATP, they're not only one primary reaction or result is to lead to these reactive oxygen species. And then that leads to lysis, and that can lead to neuroinflammation and keep this cycle going, which is bad. Another bad result of all this ATP being generated is the release of cytochrome C, which activates caspase, which can signal back to the nucleus. So remember, this is all inside the cell. Signal back to the nucleus, activate apoptotic genes, let's say, and those genes will start kicking off proteins, which will lead to programmed cell death. And this now this neuron will die through that process. So there's two ways that people often note neurodegeneration or cell death. So lysis is one, and then apoptosis is another. They're slightly different. The neurons look different. So in lysis, remember, the neuron looked like it was kind of exploded. It was kind of dashed and dashed because those reactive oxygen species work on the cell membrane. In apoptosis, it's not exactly the same. So, an apoptotic neuron just has certain features that make it look dead, and I can't really draw them out properly on this slide, but on the PowerPoint that I put up, I'll have two cortical neurons, apoptosis versus necrosis. So lysis leads to necrosis. Okay, so that's the form of cell death. So there'll be a slide up on the PowerPoint that looks at apoptosis versus necrosis. A cortical neuron, one shows necrosis, that looks like it exploded. The apoptotic neuron just looks sick. I don't. It's really, like I say, it's hard to say. The organelles are quite large. They're just not. It doesn't look like a normal, neuron. So, and also as the process of apoptosis sets off, actually that neuron will start to break into little blebs, and then you'll get phagocytosis. So you have an inflammatory or an immune response where those apoptotic neurons will start to get eaten up, which is good because they get removed. But as that apoptotic pathway or those neurons are becoming dead, dying, neurodegenerating, they are sending off signals to attract these macrophages or these immune cells. And lo and behold, that also feeds into this pathway of neuroinflammation as another common feature of neurodegeneration. So what I'll do is I'll stop here, at least for this video. So we've covered excitotoxicity, that's an increase in calcium. So I'm gonna start pushing this idea about extrasynaptic calcium. So extrasynaptic glutamate. So glutamate, that's not necessarily released in an input-dependent process. So it's floating around. So that floating around glutamate, it can result from a traumatic brain injury, a stroke, or let's say ethanol. These are going to be three of the topics that we cover coming up in the next couple of weeks. So those can produce lots of extrasynaptic glutamate that can activate, just by virtue of that process, activate those NMDA receptors, bringing calcium into that cell body. And as those calcium levels are starting to rise, The endoplasmic reticulum are trying to buffer it, needs energy. Mitochondria are generating that energy. As they're generating that energy, they are producing reactive oxygen species, hydrogen peroxide, oxygen radicals, hydroxyl radicals, and that can lead to breakdown of the cell membrane, produce lysis, lead to necrosis, and as those cells are becoming necrotic, you need something to clean that up. So you have an immune response, neuroinflammation. The second pathway is the apoptotic pathway. So those mitochondria now, as they're kicking out ATP, they're also releasing cytochrome C. Cytochrome C activates a protein called caspase, and caspase serves as a signal to activate DNA to lead to apoptotic signals, leading to programmed cell death. So when we come over here now, we have a cell that's through apoptotic pathways is being destroyed, and it's being phagocytized, right? So it's sending off signals to say we need the cell to be destroyed, or we need the cell to be chewed up. Those signals are actually leading to a neuroinflammatory response. So we'll put it on pause there, have a question, and then we'll come back to a couple different pathways. We'll talk about neuroinflammation, protein misfolding, and then dopamine. Actually, I'm going to start with dopamine because it's related to this reactive oxygen species. So more in a minute.